Hello there, and welcome to Deep Sleep Seas. I'm so glad you found your way here tonight. Maybe you had a long day, or maybe you're the kind of person who likes drifting off to the mysteries of the ocean rather than counting sheep. Either way, tonight's journey is for you. You've got nothing to do but rest. Let your mind ease, your breathing slow, and your body grow light as if the weight of the world has softened. Because tonight we aren't simply exploring the ocean's surface. We're descending to one of the strangest, most remarkable ecosystems on Earth, places where life thrives in complete darkness, powered not by the sun, but by the very chemistry of the planet itself. These are the cold seeps, hidden oases scattered across the ocean floor where ancient hydrocarbons bubble up from deep within the Earth's crust, where methane and hydrogen sulfide escape through cracks in the seafloor and nourish entire communities of creatures that have never seen sunlight, where underwater lakes of brine sit motionless on the seabed, more toxic than any poison, yet ringed with life, where giant tube worms sway gently in the current, creatures without mouths or stomachs that can live for centuries, and where tiny bacteria perform a kind of alchemy, turning deadly chemicals into the building blocks of life itself. Some of what you hear tonight may surprise you. The deep ocean holds secrets we're only beginning to understand, and cold seeps represent one of the most profound discoveries in marine science. They've changed how we think about life, about energy, about the very possibilities of existence in extreme environments. And they're still revealing new mysteries with every expedition that ventures into the deep. But before we begin, if you find these stories relaxing, consider subscribing so you don't miss the next journey into the ocean's hidden worlds. And if you're still awake enough when this is over, let me know where in the world you're listening from. Now let's descend. Let's drift down through the water column, past the sunlit zones, past the twilight depths, down to the seafloor where cold seeps pulse with quiet, ancient energy. The term cold seep is actually something of a misnomer, a name that doesn't quite capture the truth of what these places are. When scientists first discovered them, they needed a way to distinguish these sites from the hydrothermal vents that had been found just a few years earlier. Hydrothermal vents are places where superheated water, often reaching temperatures above 300 degrees Celsius, erupts from the seafloor along mid-ocean ridges. They're dramatic, violent, spectacular fountains of mineral-rich fluid shooting up into the frigid deep ocean. Cold seeps are different. The fluids that emerge from cold seeps are not actually cold. In fact, they're often slightly warmer than the surrounding seawater, sometimes by just a degree or two. But compared to the scorching temperatures of hydrothermal vents, they might as well be freezing, and so the name stuck, cold seeps. Areas of the ocean floor where fluids rich in methane, hydrogen sulfide, and other hydrocarbons seep slowly, steadily upward through cracks and fissures in the seafloor, not erupting, not gushing, seeping. Like a slow exhalation from the earth itself, these seeps can take many forms. Sometimes they appear as gentle streams of bubbles rising from the sediment, tiny spheres of methane gas wobbling upward through the water. Sometimes they manifest as thick bacterial mats coating the seafloor, white or orange or yellow carpets of microorganisms feeding on the chemicals emerging from below. Sometimes they create brine pools, underwater lakes of hypersaline water so dense it doesn't mix with the ocean above, sitting in depressions on the seabed like pools on the surface of an alien world. And sometimes they build structures, carbonate rock formations created by chemical reactions between methane and seawater, reefs and mounds that rise from the seafloor and provide hard surfaces for life to attach and grow. Cold seeps occur all over the world's oceans. They've been found in every major ocean basin, from the shallow waters just below the continental shelf to the deepest trenches. They cluster along continental margins, those sloping regions where the continents meet the deep ocean basins. They're especially common in areas of tectonic activity, where the movement of Earth's crustal plates creates the conditions for hydrocarbons to escape. The Gulf of Mexico has thousands of cold seeps, probably more than anywhere else in the world. The Pacific Northwest, from California up through Oregon and Washington, is dotted with them. They've been discovered off the coast of Japan, along the Mediterranean seafloor, in the Arctic Ocean beneath the ice, in the fjords of Norway, off the coast of New Zealand, and in the deep waters off Antarctica. The discovery of cold seeps is relatively recent in the history of oceanography. For most of human history, we had no idea these places existed. The deep ocean was simply too remote, too inaccessible. Even after we developed submarines and diving technology, the vastness of the seafloor made discoveries difficult. You could search for years and miss these sites entirely, especially since many seeps are small, isolated patches scattered across enormous expanses of sediment. The first cold seep community was discovered in 1983 by a team of scientists led by Charles Paul. They were exploring the Florida escarpment in the Gulf of Mexico using the submersible Alvin, the same vessel that had discovered hydrothermal vents just 6 years earlier. At a depth of 3,200 meters, more than 2 miles beneath the surface, they came across something entirely unexpected: clusters of tube worms, beds of mussels, all thriving in an area where cold hydrocarbon-rich brine was seeping from the seafloor. Before this discovery, scientists believed that chemosynthetic communities, ecosystems based on chemical energy rather than sunlight, were confined to hydrothermal vents. The vents seemed unique, isolated pockets of life in an otherwise barren deep sea. But the discovery of cold seeps changed everything. Here was proof that chemosynthesis could occur in many different settings, that the deep ocean floor was far more complex and alive than anyone had imagined. Since 1983, cold seeps have been discovered at an accelerating pace. Every few months, it seems, new seeps are found, new communities documented, new species described. The ocean is revealing itself slowly, but the revelations are profound. The chemistry of cold seeps begins deep beneath the seafloor, in layers of sediment that have been accumulating for millions of years. When marine organisms die, they sink to the bottom of the ocean. Their bodies, rich in organic matter, are buried by subsequent layers of sediment. Over time, this organic material is buried deeper and deeper, subjected to increasing pressure and in some cases increasing temperature. In this environment, the organic matter begins to break down. There are two main pathways by which methane forms in marine sediments. The first is biological. Microorganisms called methanogens, a type of archaea, consume the organic matter and produce methane as a waste product. This biogenic methane formation happens relatively close to the surface of the sediment, within the first few hundred meters, where temperatures are still low. The methanogens work slowly, methodically, breaking down complex organic molecules into simpler compounds, and ultimately producing methane and carbon dioxide. The second pathway is thermogenic. When organic matter is buried very deep, kilometers below the seafloor, it experiences high temperatures and pressures. Under these conditions, the organic molecules are literally cooked, broken apart by heat into smaller hydrocarbon molecules. This process produces not just methane but also larger hydrocarbon compounds like ethane, propane, and butane, the same compounds found in natural gas and petroleum. Thermogenic methane typically forms at depths where temperatures exceed about 100 degrees Celsius. Whether biogenic or thermogenic, once methane is produced, it begins to migrate upward. Methane is lighter than water, and it's also under considerable pressure from the weight of the overlying sediment and seawater. It moves through the sediment along pathways of least resistance, through porous rock layers, along fault lines, through fractures created by tectonic activity. This migration can take thousands or even millions of years. The methane rises slowly, inexorably, following the architecture of the subsurface geology. As the methane migrates upward, it encounters changing conditions. Temperature drops, pressure changes, and at a certain combination of low temperature and high pressure, something remarkable happens. The methane can combine with water to form methane hydrate, also known as methane ice. Despite its name, methane hydrate isn't actually ice in the traditional sense. It's a clathrate, a cage-like structure where molecules of methane become trapped within a lattice of water molecules. The water molecules arrange themselves into a crystalline structure with spaces inside, and methane molecules sit in those spaces, held in place but not chemically bonded to the water. Methane hydrates are stable only under specific conditions, generally at temperatures below about 10 degrees Celsius and pressures corresponding to water depths greater than about 300 to 500 meters. These conditions are common in the deep ocean and in permafrost regions on land. The result is that enormous quantities of methane are locked up in hydrate form beneath the seafloor. Scientists estimate that there may be more methane stored in ocean floor hydrates than in all conventional natural gas, oil, and coal deposits combined. The amount is staggering, perhaps thousands of gigatons of carbon. Methane hydrate has an otherworldly appearance. It looks like dirty ice or compacted snow, white or pale gray, and it's remarkably stable when left undisturbed. But bring a sample to the surface, where pressure drops and temperature rises, and it begins to fizz and bubble as the methane escapes from its cage. You can actually light methane hydrate on fire, creating the surreal sight of ice that burns. Scientists sometimes call it fire ice, a substance that seems to defy the normal categories we use to understand matter. At cold seeps, methane hydrate often appears as exposed mounds or outcrops on the seafloor. In the Gulf of Mexico, researchers have found methane hydrate iceworms, tiny pink polychaetes that live in dense colonies directly on the hydrate, grazing on bacteria that grow on the surface. These worms are exquisitely adapted to life on what is essentially frozen methane, a substance that would be deadly to most organisms. They can't survive at normal ocean temperatures. Bring them into warmer water and they simply fall apart. But in the frigid deep, on their mounds of methane ice, they thrive. Not all the methane remains trapped in hydrate form. Some continues to migrate upward until it reaches the seafloor, where it escapes into the water column. This is where cold seeps become visible, where the chemistry of the deep meets the biology of the seafloor. Methane bubbles rise from the sediment, streams of tiny spheres wobbling upward. Dissolved methane permeates the sediment-poor water, creating a gradient that extends upward toward the seafloor surface. And in some locations, the fluids don't just seep, they flow, carrying not only methane, but also hydrogen sulfide, higher hydrocarbons, and sometimes brine. Brine formation adds another layer of complexity to cold seeps. In certain regions, particularly in the Gulf of Mexico and the Red Sea, there are massive deposits of salt buried beneath the seafloor. These salt deposits formed millions of years ago when ancient seas evaporated, leaving behind thick layers of salt and evaporite minerals. Over time, these salt layers were buried by sediment. But salt is less dense than rock, and under pressure, it can flow almost like a very slow liquid. Salt tectonics. The movement and deformation of these buried salt layers creates complex structures in the subsurface. When seawater or pore water comes into contact with these salt deposits, the salt dissolves, creating a highly concentrated brine solution. This brine is much denser than normal seawater, sometimes 3 to 8 times more salty. When this brine is expelled at the seafloor, it doesn't rise or mix easily with the overlying water. Instead, it flows downslope like a heavy liquid, following the topography of the seafloor, pooling in depressions and low areas. The result is a brine pool, an underwater lake of hypersaline water sitting on the ocean floor. Brine pools are among the most surreal features of cold seeps from the perspective of a remotely operated vehicle. A brine pool looks exactly like a lake, complete with a clearly defined shoreline and surface. You can see the boundary between the brine and the normal seawater above it, a shimmering interface where the two fluids of different densities meet. The brine is so dense that objects sink to its surface rather than passing through. A remotely operated vehicle approaching a brine pool must be careful. If it descends too far, it can become trapped in the brine, unable to generate enough thrust to escape. The brine itself is hostile to most life. The extreme salinity disrupts cellular processes. The anoxic conditions, the complete absence of oxygen, make respiration impossible for most organisms, and many brine pools also contain high concentrations of hydrogen sulfide and other toxic compounds. An animal that falls into a brine pool typically dies within minutes, experiencing cerebral hypoxia as it tries to breathe an environment that offers no oxygen. The dead animals don't decompose normally because there's little oxygen to support decomposing bacteria. Instead, they become pickled, preserved in the brine like specimens in formaldehyde. Researchers exploring brine pools often find them littered with the bodies of crabs, fish, and other creatures that made the fatal mistake of entering the brine. One particularly famous brine pool in the Gulf of Mexico has been nicknamed the Hot Tub of Despair, discovered in 2015 during an expedition studying cold seeps. This brine pool sits at a depth of about 1,000 meters. It's roughly 100 feet in circumference and 12 feet deep, an oval depression filled with water that reaches temperatures of about 19 degrees Celsius, notably warmer than the surrounding seawater at about 4 degrees Celsius. The name comes from the grim discovery that the pool is surrounded by the preserved remains of animals that fell in and died. Yet even in this deadly environment, life finds a way. The edges of brine pools, the shoreline where the brine meets normal seawater, often teem with life. Mussels form dense aggregations along the rim, their shells encrusting the margins of the pool. Bacterial mats in vivid colors, whites and yellows and oranges, coat the sediment. Small crustaceans pick their way carefully along the edge, feeding on the bacteria and detritus. These creatures are living on the boundary between two worlds, drawing energy from the chemicals in the brine while remaining in water they can breathe. It's a high-wire act, a delicate balance that supports some of the most productive ecosystems in the deep sea. The key to understanding how life thrives at cold seeps lies in a process called chemosynthesis. For most of Earth's history, scientists assumed that all life ultimately depended on photosynthesis, the process by which plants and algae and cyanobacteria use sunlight to convert carbon dioxide and water into sugars. Photosynthesis is the foundation of nearly every ecosystem on the planet's surface. Herbivores eat the plants, carnivores eat the herbivores, decomposers break down the dead and return nutrients to the soil. But it all starts with sunlight captured by chlorophyll. In the deep ocean, there is no sunlight. Below about 1,000 meters, the darkness is absolute and permanent. No photosynthesis can occur. For a long time, scientists believed this meant the deep sea must be a barren desert, a place where only a few scavengers survived by feeding on the organic matter that drifted down from the sunlit surface waters above. And indeed, much of the deep sea floor is relatively sparse, with low densities of life eking out existence on the slow rain of detritus from above. But then in 1977, scientists exploring the Galapagos Rift with the submersible Alvin discovered hydrothermal vents and the astonishing communities of life surrounding them. Giant tubeworms, huge clams, swarms of white crabs, beds of mussels, all clustered around the vents where superheated water poured from the seafloor. The density of life was staggering, greater than in many coral reefs. How could this be? What were these animals eating? The answer came from a graduate student named Colleen Cavanaugh, who proposed that chemosynthetic bacteria were the foundation of these ecosystems. Just as plants use sunlight as an energy source to build organic molecules, these bacteria were using chemical energy. Specifically, they were oxidizing hydrogen sulfide, a compound abundant in hydrothermal vent fluids, and using the energy released by this reaction to fix carbon dioxide into organic compounds. The bacteria were essentially acting like plants, but instead of sunlight, they ran on chemistry. This was a revolutionary idea. It meant that life didn't require sunlight. It could thrive anywhere there was a source of chemical energy. Anywhere reduced compounds like hydrogen sulfide or methane came into contact with oxidized compounds like oxygen or sulfate. Chemosynthesis opened up entire new possibilities for where life might exist, not just on Earth but potentially on other worlds. When cold seeps were discovered a few years later, scientists realized immediately that the same principles applied. The methane and hydrogen sulfide seeping from the seafloor could support chemosynthetic bacteria, which in turn could support entire ecosystems. The specific chemistry differs between hydrothermal vents and cold seeps. The temperatures are very different, and the longevity of the sites varies. But the fundamental principle is the same. Chemical energy replaces solar energy, chemosynthesis replaces photosynthesis, and life finds a way. The chemosynthetic process at cold seeps begins with the smallest organisms, bacteria and archaea. These microorganisms are the true foundation of the ecosystem, the primary producers that convert inorganic compounds into organic matter that can fuel the food web. Different groups of microorganisms specialize in different chemical reactions, each one extracting energy from a specific transformation. Some bacteria oxidize methane. They take methane, a reduced form of carbon with a lot of chemical energy stored in its bonds, and they oxidize it, ultimately converting it to carbon dioxide. This reaction releases energy, and the bacteria capture that energy to power the synthesis of organic molecules from carbon dioxide. It's a remarkable feat of biochemistry, essentially running photosynthesis in reverse, using chemical energy instead of light energy. Other bacteria oxidize hydrogen sulfide. Hydrogen sulfide is toxic to most life, but these bacteria have evolved enzymes that can safely handle the compound. They oxidize the sulfur, converting hydrogen sulfide to elemental sulfur or to sulfate, and they use the energy released to fix carbon. If you look closely at cold seeps, you often see white bacterial mats. thick growths of sulfur-oxidizing bacteria coating the seafloor around seep sites. These mats can be several centimeters thick, dense accumulations of cells feeding on the upwelling hydrogen sulfide. The microorganisms at cold seeps also include archaea, a domain of life distinct from both bacteria and eukaryotes. Some of the most important methane-consuming organisms at cold seeps are archaea that perform anaerobic oxidation of methane, a process abbreviated as AOM. These archaea live in the sediment beneath the seafloor, in zones where methane from below meets sulfate from the seawater above. They oxidize methane not with oxygen, which is absent in the sediment, but with sulfate, a compound dissolved in seawater. It's a strange and complex process, one that wasn't even confirmed to exist until the late 1990s, and it requires the cooperation of two different types of microorganisms working together in intimate association. The anaerobic oxidation of methane is critically important for regulating the amount of methane that escapes from the seafloor into the ocean and potentially into the atmosphere. Methane is a potent greenhouse gas, with a warming potential many times greater than carbon dioxide. If all the methane seeping up through marine sediments were to reach the atmosphere, it could have significant impacts on global climate. But the archaea consuming methane in the sediment form what scientists call the benthic filter, a biological barrier that captures and oxidizes much of the methane before it can escape. The efficiency of this benthic filter varies depending on the rate of methane seepage. At seeps with slow, steady flow, the microorganisms can consume 80% or more of the methane. But at seeps with rapid flow, or where methane bubbles rise directly through the sediment without passing through microbial zones, much more methane escapes. The balance between methane production, microbial consumption, and atmospheric release is one of the key aspects of cold seep systems, and it has implications far beyond the immediate ecosystem. The organic matter produced by chemosynthetic bacteria doesn't just benefit the bacteria themselves, it becomes the foundation for an entire food web. Some of this carbon enters the food web when other organisms simply eat the bacteria. Small grazers scrape bacterial mats from rock surfaces. Filter feeders strain bacteria from the water column. Detritivores consume bacterial clumps that slough off and settle on the sediment. But the most remarkable transfers of energy at cold seeps occur through symbiosis, through intimate partnerships between bacteria and larger organisms. The giant tube worms of cold seeps are perhaps the most iconic examples of chemosynthetic symbiosis. These creatures, members of the family Ciboglinae, are among the strangest animals in the ocean. As adults, they have no mouth, no stomach, no intestines, no anus. They cannot eat. They have no means of ingesting food in the conventional sense, and yet they grow, they reproduce. They can reach lengths of 2 meters or more. How? The answer lies in an organ called the trophosome, a spongy tissue that occupies much of the worm's body cavity. The trophosome is packed with chemosynthetic bacteria, billions upon billions of them living inside specialized cells. These bacteria are sulfur-oxidizing species that can convert hydrogen sulfide and carbon dioxide into organic compounds. The tube worm provides the bacteria with everything they need: hydrogen sulfide absorbed from the environment, carbon dioxide from seawater, and oxygen for the oxidation reaction. The bacteria, in turn, produce organic molecules, sugars and amino acids, which the tube worm absorbs and uses for its own growth and metabolism. It's a perfect arrangement. The bacteria have a protected, stable environment with a constant supply of raw materials. The tube worm has a built-in food factory that requires no mouth, no digestive system, no hunting or foraging. The two organisms are so intimately connected that neither can survive without the other. The bacteria are never found living free in the environment, only inside their hosts, and the tube worms cannot obtain nutrition any other way. Tube worms begin life as tiny larvae, swimming freely in the water column. At this stage, they have a simple gut, and they can feed on small particles. But as they mature and settle on the seafloor, they undergo a remarkable transformation. The gut disappears, the mouth seals over, and they acquire their bacterial symbionts, which enter through the skin and colonize the developing trophosome. Once this transition is complete, the tube worm is committed to a sessile symbiotic existence, rooted to the seafloor, extending its bright red plume of gill tissue into the water to absorb the chemicals it needs. The plume is one of the most visually striking features of tube worms. It's bright red because it contains high concentrations of hemoglobin, the same protein that makes our blood red. But tubeworm hemoglobin has a unique property. It can bind both oxygen and hydrogen sulfide simultaneously. Normally, hydrogen sulfide would poison hemoglobin, blocking oxygen transport. But tubeworm hemoglobin has evolved special binding sites that keep the two chemicals separated, allowing the worm to transport both from the environment to the bacteria in the trophosome. It's a biochemical feat that required millions of years of evolution to perfect. Different species of tubeworms live at different types of SEEPS, and they show varying adaptations to local conditions. Some species are what researchers affectionately call short and fat, with thick bodies and relatively short tubes. These tend to live at SEEPS with high concentrations of hydrogen sulfide. Other species are long and skinny, with slender bodies extending from narrow tubes. These are often found at SEEPS with lower sulfide concentrations, where they compensate by having more surface area for absorption. The variations suggest that tubeworms have diversified to exploit different niches within the cold SEEP environment. Tubeworms at cold SEEPS can live for extraordinarily long periods. One species, Lamellabrachia limesi, has been estimated to live for more than 250 years. Some individuals might even reach 300 years or older. This makes them among the longest-lived invertebrates on Earth, comparable in lifespan to giant clams and certain deep-sea sponges. Their longevity likely reflects the stability of cold SEEPS, which can remain active for thousands or even tens of thousands of years, as well as the slow metabolism typical of deep-sea organisms. The growth of these tubeworms is correspondingly slow. They might grow only a few centimeters per year, adding new sections to their tubes gradually over decades and centuries. The tubes themselves are made of chitin, the same material that forms insect exoskeletons and fungal cell walls. The tubeworm secretes the chitin from specialized glands, building up the tube layer by layer. Old tubes can become quite thick and rigid, providing strong protection against predators and mechanical damage. When tubeworms die, their tubes remain standing on the seafloor for years, gradually filling with sediment or becoming colonized by other organisms. Mussels are the other major group of large animals at cold SEEPS that rely on chemosynthetic symbiosis. Like tubeworms, cold SEEP mussels harbor bacteria in their gill tissues. But unlike tubeworms, mussels retain the ability to feed by filtering particles from the water. They're called mixotrophs, organisms that can obtain energy through multiple pathways. When bacterial production is high, they rely primarily on their symbionts. When particle concentrations in the water are elevated, they supplement their diet by filter feeding. This flexibility may be one reason mussels are so successful at cold SEEPS. The bacteria living in cold SEEP mussels are typically methane oxidizers rather than sulfide oxidizers, though some mussel species host both types. The methane oxidizing bacteria, called methanotrophs, consume methane dissolved in the seawater and use it to produce organic compounds. The mussels position themselves in areas where methane concentrations are high but not so high that oxygen is depleted. They pump water through their gills, extracting both methane for their symbionts and oxygen for respiration. Mussel beds at cold SEEPS can be astonishingly dense. In some locations, mussels cover the seafloor in aggregations that extend for tens or even hundreds of meters. The individual mussels pack together so tightly that their shells overlap, creating a living pavement. These beds often develop distinct zones. The inner zone, closest to the center of the SEEP where methane flux is highest, typically has the largest mussels with the best growth rates. The outer zone, where methane concentrations are lower, has smaller mussels that grow more slowly. The zonation reflects gradients in resource availability, with the most successful individuals claiming the best real estate. Mussels modify their environment in ways that benefit the broader ecosystem. Their shells provide hard substrate in areas that would otherwise be soft sediment. Other organisms settle on the mussel shells, creating a three-dimensional habitat structure. Small worms bore into the shells. Barnacles attach to them. Anemones nestle between them. The mussel bed becomes a foundation for biodiversity, a refuge and resource for dozens of other species. In this way, mussels act as ecosystem engineers, organisms whose presence fundamentally alters the physical and biological characteristics of their environment. The cold SEEP clams are another group of symbiont-bearing bivalves. These clams, belonging to the family Vesicomidae, can grow quite large, with some species reaching shell lengths of more than 20 centimeters. Like mussels, they harbor sulfur-oxidizing bacteria in their gills. But clams differ from mussels in their lifestyle. Clams burrow into the sediment, extending a long muscular foot down into the anoxic, sulfide-rich layers below the surface. The foot acts like a root system, absorbing hydrogen sulfide from the sediment-poor water and transporting it up to the bacteria in the gills. This lifestyle allows clams to access hydrogen sulfide that never reaches the seafloor surface, exploiting a resource that would otherwise be unavailable. It also means clams can live in areas where seepage is too diffuse to support dense bacterial mats or mussel beds. The clams are there, hidden beneath the sediment, their siphons barely visible at the surface, quietly farming hydrogen sulfide from below. Clam beds can persist for decades or centuries, slowly expanding as larvae settle and grow. The dead shells of previous generations accumulate, creating shell hash that alters the sediment texture and provides habitat for other organisms. Fossil cold seeps preserved in ancient rocks often contain abundant clam shells, testament to the longevity and productivity of these ecosystems across geological time. Beyond the symbiont-bearing megafauna, cold seeps host a diverse array of other organisms. Polycate worms of many species burrow through the sediment or build tubes on hard surfaces. Some are deposit feeders, consuming sediment and extracting organic matter. Others are carnivores, hunting small prey among the mussels and bacterial mats. Still others have their own symbiotic bacteria, forming yet another pathway for chemosynthetic energy to enter the food web. Crustaceans are abundant at cold seeps. Small amphipods swarm over bacterial mats, grazing on the microorganisms. Larger crabs patrol the seep sites, scavenging and predating. Some crab species seem to be endemic to cold seeps, found nowhere else. They've evolved physiological adaptations to tolerate the high concentrations of hydrogen sulfide and the low oxygen levels that often characterize seep environments. Fish visit cold seeps, drawn by the abundance of invertebrate prey. Crenideers, also called rat-tails, are common deep-sea fish that forage around seeps. Eelpouts, thick-bodied fish with blunt heads, are also frequently seen. These fish are typically not specialists. They don't depend exclusively on seep ecosystems, but they recognize seeps as productive areas where food is more abundant than on the surrounding seafloor, and they time their visits accordingly. Octopods occasionally appear at cold seeps, ghostly pale animals that hunt among the mussels for crabs and worms. Their movements are slow and deliberate, conserving energy in the food-poor deep sea. When they find a good hunting ground like a cold seep, they may remain in the area for days or weeks, methodically working through the available prey before moving on. Even animals that seem entirely unrelated to chemosynthesis can benefit from cold seeps indirectly. Rockfish and other commercially important species in the Pacific Northwest use cold seep areas as essential habitat, particularly the carbonate rock formations that develop over time at seep sites. These hard structures provide refuge and spawning habitat in regions where most of the seafloor is soft sediment. The seeps thus support not only chemosynthetic specialists but also a broader community of organisms that simply take advantage of the habitat complexity. The carbonate formations at cold seeps are themselves products of chemosynthesis, specifically of the anaerobic oxidation of methane. When archaea oxidize methane in the sediment, the reaction produces bicarbonate ions. These ions combine with calcium dissolved in seawater to precipitate calcium carbonate, the mineral that makes up limestone. The precipitation occurs within the sediment, cementing the particles together. Over time, these cemented layers build up, forming solid rock that can outcrop at the seafloor surface. The carbonate rocks at cold seeps have a distinctive appearance. They're often porous and irregularly shaped, with cavities and channels where fluids once flowed. They may be stained orange or brown from iron compounds, or white from microbial precipitates. The rocks provide the hard substrate that tubeworms and other sessile organisms require for attachment. Without the carbonate formations, many seeps would be unable to support dense populations of the large, conspicuous animals that characterize these ecosystems. In some locations, carbonate precipitation has been occurring for thousands of years, building massive structures. Some cold seep sites feature carbonate mounds several meters tall and tens of meters across, complex three-dimensional habitats that rise above the surrounding seafloor. These mounds can persist even after seepage has ceased, providing long-lasting habitat for organisms that don't depend on active chemosynthesis. In this way, cold seeps leave a legacy, structuring the seafloor long after the methane has stopped flowing. Ancient cold seeps provide a window into Earth's past. When sediments containing cold seep carbonates are uplifted and exposed on land, they preserve a record of ancient seep ecosystems. Geologists have found fossil cold seeps in rocks that are tens or even hundreds of millions of years old. These fossil seeps contain the remains of chemosynthetic communities, including fossil tube worms, clams, and mussels that are the ancestors of modern seep fauna. The existence of ancient seeps tells us that chemosynthetic ecosystems have been a feature of Earth's oceans for a very long time, probably for as long as there has been a seafloor and organic matter being buried and decomposing. The specific animals inhabiting seeps have changed over evolutionary time, but the fundamental ecology, the dependence on hydrocarbon seepage and chemosynthetic bacteria, appears to be ancient. Life has been exploiting the chemical energy of the deep Earth for hundreds of millions of years, a parallel universe of energy flow that existed long before humans walked the planet. The broader significance of cold seeps extends beyond their immediate ecology. These ecosystems are intimately connected to the global carbon cycle and to climate. Methane, the primary hydrocarbon seeping from the seafloor, is a powerful greenhouse gas. Molecule for molecule, methane traps about 25 times more heat in the atmosphere than carbon dioxide over a 100-year period. In the short term, over 20 years, methane is about 80 times more potent. This means that even relatively small amounts of methane can have large climatic effects. Most of the methane seeping from cold seeps never reaches the atmosphere. The microorganisms in the sediment and at the seafloor consume it, oxidizing it to carbon dioxide before it can escape into the water column. And even the methane that does enter the water column is typically oxidized by free-living bacteria long before it reaches the surface. Studies of methane budgets suggest that less than 10% of the methane seeping from the seafloor ultimately reaches the atmosphere. The rest is captured by the benthic filter and the water column processes. But the efficiency of this biological filter could change. Ocean warming is altering deep sea temperatures, and warmer temperatures could accelerate methane release from hydrates or reduce the efficiency of microbial methane oxidation. Changes in ocean circulation could affect oxygen concentrations at the seafloor, which would also impact methane consumption rates. There's concern among climate scientists that increasing ocean temperatures could trigger the destabilization of methane hydrate deposits, leading to large-scale methane release that could amplify global warming. This scenario is sometimes called the methane time bomb, or the clathrate gun hypothesis. The idea is that a warming ocean could melt methane hydrates in shallow marine sediments, releasing large amounts of methane that overwhelm the biological filter and escape into the atmosphere, causing rapid warming that in turn triggers more hydrate melting, creating a runaway feedback loop. Some scientists have proposed that massive methane releases from hydrates might have occurred in Earth's past, potentially contributing to rapid warming events like the Paleocene-Eocene thermal maximum about 56 million years ago. The evidence for these past methane releases remains debated, and the risk of a near-term methane catastrophe from marine hydrates is considered low by most researchers. The ocean is vast, microbial methane consumption is efficient, and most hydrate deposits are in deep, cold water, where they're stable even under moderate warming scenarios. But the concern is not zero. Cold seeps and the methane they release represent a significant term in the global carbon budget, and understanding how these systems will respond to environmental change is an active area of research. Beyond climate, cold seeps matter for marine biodiversity. These ecosystems are hotspots of endemism, places with high concentrations of species found nowhere else. Many of the tube worms, clams, and mussels at seeps are specialists that occur only in chemosynthetic environments. They've evolved over millions of years to exploit a very specific ecological niche, and they're exquisitely adapted to conditions that would be lethal to most marine life. Protecting cold seeps means protecting unique evolutionary lineages, species that represent natural experiments in adaptation to extreme environments. Cold seeps also serve as essential habitat for commercially important fish species. The carbonate structures at seeps provide hard substrate in regions where it's otherwise scarce. Many fish species require hard bottom for spawning or as juvenile habitat. In the Pacific Northwest, cold seeps have been designated as essential fish habitat under federal fisheries management regulations, recognizing that these sites play a critical role in supporting fish populations. The economic value of cold seeps extends to potential biotechnology applications. The microorganisms living at seeps possess unique enzymes and metabolic pathways adapted to function under high pressure, low temperature, and exposure to toxic chemicals. These enzymes have potential applications in industrial processes, pharmaceuticals, and bioremediation. Researchers have begun exploring the microbial communities at cold seeps as sources of novel biomolecules, a practice sometimes called bioprospecting. The discovery of new species and new biochemistry at seeps continues to yield insights with practical applications. Studying cold seeps requires sophisticated technology. The depth at which most seeps occur, typically hundreds to thousands of meters below the surface, puts them beyond the reach of conventional scuba diving. Instead, scientists rely on remotely operated vehicles, or ROVs, tethered robots equipped with cameras, manipulator arms, and sampling tools. The ROV is controlled from a ship at the surface, with pilots guiding it through the water column and across the seafloor using video feeds and sensor data. Finding cold seeps in the vastness of the ocean is a significant challenge. Early discoveries were often serendipitous, the result of submersible pilots happening upon unusual communities during exploratory dives. But modern seep science uses more systematic approaches. Multibeam sonar can map the seafloor in high resolution, revealing features like carbonate mounds or pockmarks that suggest seepage. Water column sonar can detect methane bubble streams rising from the seafloor, pinpointing active seep sites even when the seafloor features are subtle. Once a seep is located, ROVs conduct detailed surveys, documenting the fauna, collecting samples of organisms and fluids, and deploying instruments to measure chemical concentrations and flow rates. Scientists bring back sediment cores to analyze the microbial communities and the geochemistry of the subsurface. They sequence DNA from bacteria and archaea to understand which organisms are present and what metabolic processes they're performing. They measure growth rates of tube worms and mussels by tagging individuals and returning to the same sites years later. Slowly, dive by dive, sample by sample, a picture emerges of how these ecosystems function. Recent expeditions have revealed just how common cold seeps are. In 2016, a survey along the U.S. Atlantic margin used advanced sonar to map methane seeps from North Carolina to Massachusetts. The researchers identified more than 500 seep sites, the majority of which had never been documented before. This was 4 times the number previously known from the region. The discovery suggested that cold seeps are far more widespread than scientists had realized, and that large portions of the ocean floor remain unexplored. Similar findings have come from other regions. Off the coast of Washington and Oregon, surveys of the Cascadia margin have identified more than 900 methane seep sites. In the Arctic, expeditions are finding seeps along the continental margins, and even in areas that were until recently covered by sea ice year-round. As the Arctic warms and ice cover diminishes, new seep sites are being discovered, some of which may be newly active as a result of changing conditions. The discoveries continue. In 2020, researchers announced the finding of cold seeps in the Gulf of Aqaba in the Red Sea, extending the known range of seeps in that region. In 2021, an expedition to the waters off Argentina documented cold seeps along the continental margin, the first comprehensive study of seeps in the southwestern Atlantic. Every year brings new findings, new sites, new species, new insights into the workings of these systems. The exploration of cold seeps is in many ways still in its early stages. We've known about these ecosystems for only about 40 years, a tiny fraction of the history of oceanography. Large areas of the deep ocean remain completely unexplored. There are likely thousands of cold seeps that have never been visited, never been mapped, never been studied. And with each new seep that's discovered, there's potential for new species, new microbial metabolisms, new ecological interactions. Some of the most exciting frontiers in cold seep research involve understanding the connections between seeps and other deep-sea ecosystems. Scientists have known for a while that animals living at seeps and hydrothermal vents can disperse over large distances, with larvae traveling hundreds or thousands of kilometers in ocean currents. But how do larvae find new seep sites? How do populations maintain genetic connectivity across vast distances? These questions touch on fundamental issues in ecology and evolution. One hypothesis is that there are stepping stones between chemosynthetic habitats, intermediate sites, that allow larvae to hop from one location to another. These stepping stones might include whale falls, the carcasses of dead whales that sink to the seafloor and create temporary islands of organic enrichment. As a whale decomposes, it releases hydrogen sulfide, and chemosynthetic bacteria colonize the bones. Some of the same species of mussels and clams that live at seeps and vents also appear at whale falls, suggesting these ephemeral habitats play a role in connectivity. Other potential stepping stones include wood falls, logs, and other plant material that wash into the ocean and sink. Certain deep-sea wood-boring bivalves harbor bacterial symbionts and can live on sunken wood. When the wood is exhausted, larvae from these populations might disperse to nearby seeps. The deep ocean, in this view, is a mosaic of chemosynthetic habitats, some long-lived like seeps, others ephemeral like whale falls, but collectively forming a network that allows specialized species to persist across geological time. Cold seeps may also have implications for the search for life beyond Earth. One of the key findings from studying chemosynthetic ecosystems is that life doesn't require sunlight. It can thrive anywhere there's a source of chemical energy and the basic ingredients for biochemistry. This expands the range of environments where we might expect to find life in the universe. Jupiter's moon, Europa, for example, is covered in ice, but beneath the ice is thought to be a global ocean of liquid water. Tidal heating from Jupiter's gravity keeps the interior warm enough to maintain liquid water, and there may be hydrothermal vents on Europa's seafloor. If so, Europa could have chemosynthetic ecosystems, analogous to those at Earth's hydrothermal vents or cold seeps. The methane and hydrogen detected in plumes erupting from Europa's surface suggest that the chemistry might be right. Life on Europa, if it exists, would almost certainly be microbial, perhaps similar to the bacteria and archaea at Earth's cold seeps. Saturn's moon Enceladus is another candidate. Enceladus also has a subsurface ocean, and plumes of water vapor erupting from the south polar region contain methane, hydrogen, and organic molecules. The chemistry of these plumes is consistent with hydrothermal activity and possibly with microbial methanogenesis. The discovery of chemosynthetic ecosystems on Earth has made worlds like Europa and Enceladus prime targets in the search for extraterrestrial life. Even Mars, now a frozen desert, might once have had subsurface environments where chemosynthesis could occur. Evidence suggests that Mars once had liquid water on its surface, and there may still be liquid water in deep aquifers or beneath ice caps. If Mars harbored life in its past, or if microbial life persists in the subsurface today, it would likely be chemosynthetic, living off the chemical energy available in rock-water interactions. The principles we've learned from studying cold seeps and hydrothermal vents inform how we think about Martian habitability. Back here on Earth, cold seeps continue to reveal their secrets. Researchers are investigating the full diversity of microbial life at seeps, using metagenomic approaches to sequence the DNA of entire communities. They're discovering new groups of microorganisms, new metabolic pathways, new symbioses. The microbial world is vastly more diverse than the visible megafauna, and we're only beginning to catalog it. Scientists are also studying how seep ecosystems change over time. Seeps are dynamic, not static. The rate of methane flux can vary seasonally or over longer cycles. Some seeps pulse with periods of high activity followed by quiescence. Others remain stable for decades or centuries. Understanding these dynamics helps predict how seeps will respond to environmental change, including ocean warming and acidification. There's also growing interest in the role of cold seeps in supporting fisheries. If seeps provide essential habitat for commercially important species, then any activity that damages seeps could have economic consequences. This includes deep-sea fishing practices that disturb the seafloor, such as bottom trawling. It also includes oil and gas development, since drilling activities can intersect seep sites or alter subsurface fluid flow. Balancing resource extraction with ecosystem protection is an ongoing challenge in ocean management. The medical and pharmaceutical potential of cold seep microorganisms is another area of active research. Some of the bacteria and archaea at seeps produce unusual compounds as part of their metabolism, compounds that might have antimicrobial, antiviral, or anti-cancer properties. The extreme conditions at SEEPS select for biochemical pathways that don't exist in terrestrial organisms, and these pathways can be sources of novel drugs or industrial enzymes. One of the most fascinating aspects of cold SEEPS is the sheer scale of the methane reservoir they represent. The amount of methane stored in hydrate form beneath the ocean floor is enormous, possibly exceeding all conventional fossil fuel reserves combined. This has led to interest in methane hydrates as a potential energy source. Countries like Japan, which have limited domestic energy resources, are exploring technologies to extract methane from hydrate deposits. Extracting methane from hydrates is technically challenging. The hydrates are stable only under high pressure and low temperature, so bringing them to the surface causes them to dissociate, releasing methane gas. Controlling this dissociation and capturing the gas without losing it to the water column or causing seafloor instability is difficult. There are also environmental concerns. Large-scale extraction could destabilize the seafloor, potentially triggering submarine landslides. And releasing large amounts of methane, even if most is captured, could have climatic impacts. For now, methane hydrates remain largely unexploited, but the technology is advancing, and it's possible that hydrates will play a role in future energy systems. If so, understanding cold seeps, the natural analog of hydrate systems, will be critical for managing extraction sustainably. The study of cold seeps also contributes to our understanding of Earth's geological history. Methane seepage has been occurring for hundreds of millions of years, possibly for billions. The methane cycle is intimately linked to the carbon cycle, to plate tectonics, to the evolution of life. By studying modern seeps, we gain insights into how Earth systems have operated over geological time. For instance, there's evidence that major extinction events in Earth's past may have been associated with methane releases. The Permian-Triassic extinction, the worst mass extinction in Earth's history, coincided with evidence of massive methane release and ocean anoxia. Some researchers propose that volcanic eruptions triggered the melting of methane hydrates, leading to runaway warming and ocean acidification that killed most marine life. The hypothesis remains contentious, but it highlights the potential for methane to play a role in major environmental upheavals. Cold seeps are places of beauty, though it's a strange, alien beauty that bears little resemblance to the vibrant colors of coral reefs or the gentle sway of kelp forests. The beauty of a cold seep lies in its improbability, in the tenacity of life clinging to chemical gradients in absolute darkness. The sight of a dense bed of tube worms swaying in the current, their red plumes extended, is a reminder that life finds a way, that evolution is endlessly creative, that the boundaries of habitability are wider than we once imagined. As remotely operated vehicles have brought back images and video from more and more seep sites, our appreciation for these ecosystems has grown. The videos show fields of mussels so dense they pave the seafloor. Bacterial mats in vivid white and orange rippling gently in bottom currents, shimmering brine pools with defined shorelines, surrounded by halos of life, carbonate mounds rising like miniature mountains from the sediment, tube worms clustered around vents, their plumes filtering the water. It's a world utterly different from anything on land, yet it follows the same fundamental principles of ecology, energy flow, and evolution. The discoveries at cold seeps have humbled us, reminding us how much we still have to learn about our own planet. The deep ocean remains one of the least explored environments on Earth. We've mapped the surface of Mars in higher resolution than much of our own seafloor, and yet each time we send an ROV to the deep sea, we find something new, something unexpected. Cold seeps are part of that larger story of discovery, a reminder that Earth still holds mysteries, that there are still frontiers to explore, still questions to answer. Consider the diversity of life forms that inhabit these ecosystems. Beyond the iconic tube worms and mussels, there exists a remarkable array of smaller organisms, each playing crucial roles in the seep community. The polychaete worms that burrow through the sediment come in dozens of species, each with specialized feeding strategies. Some are deposit feeders, processing sediment particles and extracting whatever organic matter they contain. Their burrows ventilate the sediment, creating pathways for oxygen to penetrate deeper into the substrate, which in turn affects microbial activity and chemical gradients. Other polychaetes are suspension feeders, extending feathery appendages into the water column to capture bacteria and particulate organic matter. They position themselves strategically, often near tubeworm bushes or at the edges of mussel beds, where the concentration of suspended material is highest. Their feeding helps clarify the water, removing bacteria and particles that might otherwise settle out and smother the seafloor. Carnivorous polychaetes patrol the surfaces of rocks and shells hunting for smaller prey. Some have fearsome jaws capable of capturing and consuming other worms, small crustaceans, and even juvenile bivalves. These predators help regulate population densities of their prey species, preventing any single group from dominating the available space. The amphipods and other small crustaceans that live among cold seep communities are equally diverse. Some species are scavengers, feeding on dead tubeworms, muscle tissue, and other organic detritus. When a tubeworm dies, its body is quickly colonized by these scavengers, which break down the tissues and recycle the nutrients back into the ecosystem. Without these decomposers, dead organic matter would accumulate, potentially creating anoxic zones that would be uninhabitable for most organisms. Other amphipods graze on bacterial mats, cropping the microorganisms like tiny underwater gardeners. Their grazing can actually stimulate bacterial growth in some cases, much like how grazing mammals on land can promote plant growth by removing old tissue and recycling nutrients. The relationship between grazers and bacteria at cold seeps is complex, involving feedback loops that scientists are still working to understand. The fish communities around cold seeps are more diverse than was initially appreciated. Early observations suggested that fish were merely visitors, passing through seep sites in search of food, but longer-term studies using baited cameras and acoustic tags have revealed that some fish species spend considerable time at seeps, returning repeatedly to the same sites. These fish may be drawn not just by abundant prey, but also by the physical structure of the seep environment. The carbonate rocks and mussel beds at seeps create complex three-dimensional habitats that are rare in the deep sea. Most of the deep ocean floor is relatively flat and featureless, covered in soft sediment that offers little in the way of shelter or attachment sites. The hard structures at seeps provide refuge from predators, spawning sites, and places for fish to rest. Juvenile fish, in particular, may use seep structures as nursery habitat, where they can hide from larger predators while feeding on the abundant small invertebrates. Some fish species may even benefit from the altered water chemistry at seeps. The elevated carbon dioxide levels produced by microbial respiration can affect fish physiology, and some species appear to be tolerant of or even adapted to these conditions. Understanding how fish respond to the geochemical signatures of seeps could help predict how marine communities will respond to ocean acidification, which is raising carbon dioxide levels throughout the world's oceans. Sharks and rays occasionally appear at cold seep sites, ghostly shapes emerging from the darkness beyond the reach of ROV lights. Deep-sea sharks are poorly studied, and their association with cold seeps is not well understood. They may be attracted by the fish and invertebrate prey concentrated at seeps, or they may simply be passing through during their normal ranging behavior. Some shark species are known to have long-distance migration patterns covering hundreds or thousands of kilometers, and seeps might serve as waypoints or landmarks in these journeys. The microbial mats that coat so many surfaces at cold seeps are themselves miniature ecosystems of remarkable complexity. What appears to the human eye as a simple white or orange film is actually a structured community of multiple bacterial species, each occupying a specific niche within the mat. The surface layer might be dominated by sulfur-oxidizing bacteria that have direct access to oxygen in the overlying water. Beneath them, in the anoxic zones within the mat, sulfate-reducing bacteria and methanogens carry out their metabolic processes. Different layers have different pH levels, different chemical compositions, different temperatures. These stratified microbial communities are similar in some ways to microbial mats found in hot springs or hypersaline lagoons, environments where the extreme conditions exclude most large organisms but allow bacteria to thrive. The study of modern microbial mats at cold seeps and elsewhere provides insights into what early Earth ecosystems might have looked like before the evolution of plants and animals. For much of Earth's history, life was exclusively microbial, and the planet's surface was covered in bacterial and archaeal communities that shaped the chemistry of the oceans and atmosphere. The genetic diversity within cold seep microbial communities is staggering. A single sample of sediment from a seep site might contain thousands of different species of bacteria and archaea, many of which have never been cultured in a laboratory. Scientists use DNA sequencing to identify and characterize these microorganisms, constructing phylogenetic trees that show how different groups are related. These analyses have revealed entirely new branches on the tree of life, groups of organisms that were unknown until cold seeps were studied. Some of the most interesting discoveries involve microorganisms that form syntrophic relationships, partnerships where two or more species cooperate to carry out metabolic processes that neither could accomplish alone. The anaerobic oxidation of methane, for example, requires the cooperation of methanotrophic archaea and sulfate-reducing bacteria. The archaea oxidize methane to carbon dioxide, but cannot complete the process without a partner to remove electrons. The bacteria reduce sulfate to sulfide, accepting the electrons from the archaea. Together, the two organisms can extract energy from methane oxidation, but separately, neither can. These syntrophic relationships are thought to be ancient, perhaps dating back to the earlier stages of life on Earth. They represent a form of cooperation that predates the evolution of multicellular life, a reminder that symbiosis and mutualism have been central to biology since the beginning. The discovery of syntrophy at cold seeps has implications for understanding the origin of eukaryotic cells, which are thought to have originated through symbiotic mergers of different bacterial lineages. Cold seeps also host a variety of organisms that don't fit neatly into the categories of producers, consumers, or decomposers. Some organisms are mixotrophs, capable of both photosynthesis and heterotrophy, though in the deep sea they rely entirely on consuming other organisms or dissolved organic matter. Others are chemolithotrophs that derive energy from inorganic compounds but can also consume organic matter when it's available. The metabolic flexibility of many deep-sea organisms reflects the variable and unpredictable nature of resource availability in these environments. The reproductive strategies of cold seep organisms are adapted to the challenges of living in isolated, patchy habitats scattered across vast distances. Many species produce large numbers of small larvae that disperse in ocean currents, a strategy that maximizes the chances of finding new seep sites but results in high larval mortality. The larvae spend weeks or months drifting in the water column, feeding on plankton if they can, gradually sinking toward the seafloor. Most never find a suitable habitat and perish, but a few, by chance or by detecting chemical cues, locate a seep and settle there, beginning a new generation. The chemical cues that larvae use to find seeps are an active area of research. It's known that some larvae can detect hydrogen sulfide or methane in the water, and they orient toward these signals. But the concentrations must be within a certain range too low, and the larvae can't detect it, too high, and it might indicate toxic conditions. The larvae must also be able to distinguish between cold seep chemicals and those from other sources, like decomposing organic matter or hydrothermal vents. The sensory and behavioral mechanisms involved are only beginning to be understood. Once larvae settle and metamorphose into adults, their survival depends on successfully establishing themselves in the competitive environment of the seep. Space is limited, especially on hard substrates where sessile organisms like tube worms and mussels attach. Competition for space can be intense, with faster-growing species outcompeting slower ones and early arrivals monopolizing the best locations. The outcome of this competition shapes the structure of the community, determining which species are abundant and which are rare. The growth rates of cold seep organisms vary dramatically depending on the availability of energy-rich chemicals. In areas of high methane or sulfide flux, tube worms and mussels can grow rapidly, reaching large sizes within a few years. In lower flux areas, growth is much slower, and the organisms might take decades to reach maturity. This variation creates patchiness in size structure across a seep site, with zones of large, fast-growing individuals near the center of active seepage and zones of smaller, slower-growing individuals at the periphery. The longevity of individual tube worms and mussels has implications for the stability and persistence of seep communities. If individuals can live for centuries, then populations can persist even if recruitment is sporadic. A few successful larval settlement events spaced decades or centuries apart could be sufficient to maintain a population. This buffering against recruitment failure is important in environments where larval supply is unpredictable, which is likely the case for isolated seep habitats. The death of large tube worms and mussels creates disturbances within the seep community, opening up space for new colonists and altering local chemical conditions. When a tube worm bush collapses, either from physical damage or natural senescence. The sudden availability of hard substrate triggers a successional sequence. Initially, fast-growing bacteria colonize the empty tubes. Then small invertebrates move in, followed by larval mussels or juvenile tubeworms. Over time, a new community develops, potentially different in composition from the one that preceded it. These successional dynamics give SEEP communities a mosaic structure, with patches in different stages of development scattered across the site. Some areas have mature, well-established populations of large organisms. Other areas are in early stages of colonization, dominated by opportunistic species. Still other areas might be barren, recently disturbed and awaiting recolonization. This spatial heterogeneity contributes to overall biodiversity, as different species are favored in different successional stages. The temporal dynamics of cold seeps add another layer of complexity. Seeps are not static features. The flux of methane and other chemicals can vary over time, responding to changes in subsurface pressure, permeability of the sediment, or tectonic activity. Some seeps pulse on annual or decadal cycles. Others show no obvious pattern, with flux varying unpredictably. When seepage declines, the organisms dependent on it face stress. Bacterial production drops, symbiont-bearing animals receive less nutrition, and the community may contract or shift in composition. If seepage ceases entirely, the community can collapse. Tubeworms and mussels, deprived of their symbiote's food source, slowly starve. The bacteria die off. Within a few years, an inactive seep might be largely devoid of the distinctive seep fauna, though the carbonate structures remain as evidence of past activity. These extinct or dormant seeps are common on the seafloor, ghostly remnants of once thriving communities. But seeps can also reactivate. A shift in subsurface geology might reopen pathways for methane flow, and seepage resumes. If the dormant period was short, some organisms might still be present, able to take advantage of the renewed resource. If the dormant period was long, recolonization must occur from larvae dispersing from other seeps. The reactivation of an extinct seep provides an opportunity to observe community assembly from the beginning, to see which species arrive first and how the community develops over time. The study of cold seeps has also revealed unexpected connections between seeps and other deep-sea ecosystems. For example, there's evidence that some species found at seeps also occur at whale falls or wood falls, those ephemeral chemosynthetic habitats created by sunken organic material. The distributions suggest that these habitats are linked by larval dispersal, that they form a network of stepping stones, allowing chemosynthetic specialists to persist in the deep sea despite the patchiness and unpredictability of any single habitat type. The concept of a chemosynthetic network is appealing, but many questions remain. How often must larvae encounter suitable habitat to maintain population connectivity? How far can larvae disperse before their energy reserves are exhausted? Do different species have different dispersal capabilities? And how does this affect their geographic ranges? These are fundamental questions in marine ecology, and cold seeps provide a natural laboratory for addressing them. Cold seeps in different ocean basins host different communities, reflecting both evolutionary history and environmental conditions. Seeps in the Atlantic tend to have different tubeworm and mussel species than seeps in the Pacific. Mediterranean seeps have their own distinctive fauna, as do seeps in the Antarctic and Arctic. Some species have wide distributions, found at seeps across multiple ocean basins, while others are endemic to specific regions. Understanding these biogeographic patterns helps reconstruct how seep fauna have evolved and dispersed over millions of years. Fossil seeps provide a long-term perspective on these biogeographic patterns. By examining the distributions of seep fossils in rocks of different ages, paleontologists can map how seep communities have changed over geological time. Some seep lineages, like the vesicomid clams, have a fossil record extending back more than 100 million years, suggesting these groups have been associated with seeps since the Mesozoic era. Other groups appear more recently in the fossil record, suggesting more recent evolutionary origins. The fossil record also reveals that some ancient seep communities were quite different from modern ones. Certain groups of organisms that were common at seeps in the past are now extinct. Others have shifted their distributions or changed their ecological roles. The study of fossil seeps is challenging because soft tissues are rarely preserved, and identifying ancient seep sites requires recognizing the distinctive carbonate formations and chemical signatures. But when well-preserved fossil seeps are found, they provide invaluable windows into the evolutionary history of chemosynthetic life. One of the remarkable things about cold seeps is that they occur in so many different tectonic settings, each with its own geological context and hydrocarbon sources. In subduction zones, where one tectonic plate dives beneath another, sediments are compressed and fluids are squeezed out. These fluids carry methane and other hydrocarbons formed from organic matter in the subducting sediments. The seeps that form along subduction zones, like those off the coasts of Japan, Oregon, and Chile, are often associated with methane hydrates and carbonate formations. In passive margins where no active tectonic boundary exists, seeps can still form through sediment compaction and salt tectonics. The Gulf of Mexico is a passive margin, but it has some of the most active seeps in the world because of the massive salt deposits beneath the seafloor. As sediments accumulate, they compress the salt, which flows and deforms, creating fractures and pathways for fluid migration. The result is a landscape dotted with seeps, brine pools, and mud volcanoes. Transform margins, where plates slide past each other, also host seeps. The movement along fault zones can create fractures that allow hydrocarbons to escape. The seeps along transform margins tend to be linear, following the traces of faults across the seafloor. These linear seep systems can extend for kilometers, creating elongated oases of life in otherwise barren regions. Even mid-ocean ridges, those underwater mountain ranges where new oceanic crust is formed, can have cold seeps in addition to hydrothermal vents. In some ridge segments, sediments accumulate in rift valleys, and organic matter buried in those sediments produces methane. If the methane finds pathways to the surface, seeps can develop. These ridge-associated seeps are less common than vent systems, but they demonstrate that methane seepage can occur in almost any marine geological setting. The global distribution of cold seeps is still being mapped, as more of the seafloor is surveyed with advanced sonar, and as more deep-sea expeditions are conducted. The number of known seeps continues to grow. Scientists estimate there may be tens of thousands of cold seeps worldwide, perhaps more. Each represents a unique ecological experiment, a local community shaped by the specific conditions of that site: water depth, temperature, methane flux rate, sediment type, current regime, and distance from other seeps. Comparing seeps across this global distribution allows scientists to identify general principles and predict where undiscovered seeps might be found. It also allows assessment of how representative any single seep study might be. If we've only studied seeps in the Gulf of Mexico intensively, how much can we generalize to seeps in other regions? Are the fundamental ecological processes the same, or does each ocean basin have its own rules? These questions motivate efforts to study seeps in diverse locations, to build a truly global understanding of these systems. The technological advances that have enabled cold seep research are themselves a fascinating story. The development of deep-diving submersibles in the mid-20th century opened the deep ocean to direct human observation for the first time. Alvin, launched in 1964, has been continuously upgraded and remains in service today, having made more than 5,000 dives. It was Alvin that discovered the first hydrothermal vents and the first cold seeps, fundamentally changing our understanding of the deep sea. Remotely operated vehicles, or ROVs, have complemented submersibles by allowing longer dive times and access to greater depths without the risk and expense of carrying human passengers. Modern ROVs are sophisticated robots equipped with high-definition cameras, powerful lights, manipulator arms capable of delicate sampling, and suites of sensors measuring temperature, salinity, oxygen, methane, and other parameters. They can work at depths beyond the reach of human-occupied submersibles, down to the deepest trenches. Autonomous underwater vehicles, or AUVs, are another tool in the deep sea exploration toolkit. Unlike ROVs, which are tethered to a ship, AUVs operate independently, following pre-programmed missions, or responding to sensor inputs. They're particularly useful for mapping large areas of seafloor, conducting surveys that would take weeks or months with an ROV. Some AUVs are equipped with chemical sensors that can detect methane plumes in the water column, autonomously locating seeps and marking their positions for later investigation. Advances in sonar technology have revolutionized the search for cold seeps. Multibeam echo sounders can map the seafloor in fine detail, revealing subtle features like pockmarks, carbonate mounds, and sediment disturbances that indicate seepage. Water column sonar can detect bubbles rising from the seafloor, even in deep water where the bubbles would never reach the surface. By combining these sonar techniques, scientists can rapidly survey large areas and identify sites worthy of further investigation with ROVs or submersibles. Molecular techniques have transformed the study of cold seep microbiology. In the past, understanding microbial communities required culturing organisms in the laboratory, a process that works for only a small fraction of environmental microbes. Most bacteria and archaea refused to grow in artificial media, and their existence was unknown. The development of DNA sequencing technologies changed everything. By extracting DNA directly from environmental samples and sequencing it, scientists can identify organisms without culturing them, revealing the full diversity of microbial life. Metagenomics, the sequencing of all DNA in a sample, provides even deeper insights. It reveals not just which organisms are present, but what genes they carry, what metabolic capabilities they have, what roles they play in the ecosystem. Metagenomic studies of cold seep sediments have uncovered novel metabolic pathways, new forms of cooperation between organisms, and unexpected diversity in even the most extreme conditions. Stable isotope analysis is another powerful tool for studying cold seeps. Different carbon sources have different ratios of carbon-12 to carbon-13 isotopes. Methane is typically very depleted in carbon-13. So organisms that derive their carbon from methane-oxidizing bacteria have distinctive isotopic signatures. By measuring the carbon isotope ratios in tissues, scientists can trace the flow of carbon through the food web, determining which organisms rely on seep-derived carbon and which obtain their nutrition from other sources. These isotopic studies have revealed that some organisms living at seeps don't actually depend on seep production. They're simply taking advantage of the habitat structure or feeding on prey that are attracted to the seep. Other organisms show clear isotopic evidence of reliance on chemosynthetic carbon, with values that could only come from methane or hydrogen sulfide oxidation. The food webs at seeps are thus more complex than a simple linear progression from bacteria to grazers to predators. They're mosaics of multiple carbon sources and energy pathways, with different species occupying different positions in a multidimensional niche space. The relationship between cold seeps and human activities is complex and multifaceted. On one hand, seeps are indicators of hydrocarbon deposits beneath the seafloor, which makes them of interest to oil and gas industries. The presence of active seepage suggests that significant quantities of methane and other hydrocarbons exist in subsurface reservoirs, potentially accessible through drilling. In some regions, exploration companies use the locations of natural seeps to guide decisions about where to drill. The seeps are like surface expressions of deeper geological structures, windows that reveal what lies beneath. This connection between seeps and hydrocarbons means that areas with active seeps often coincide with areas of industrial interest. The Gulf of Mexico, with its thousands of cold seeps, is also one of the most intensively developed offshore oil and gas provinces in the world. Thousands of wells have been drilled, hundreds of platforms erected, and an extensive network of pipelines laid across the seafloor. This industrial activity occurs in the same regions that host seep ecosystems, creating potential conflicts between resource extraction and ecosystem protection. The impacts of oil and gas development on cold seeps are not fully understood. Drilling operations can alter subsurface pressures and fluid flow pathways, potentially affecting natural seepage patterns. Seeps might increase or decrease in activity, or new seeps might form. The placement of infrastructure like pipelines and platforms can physically damage seep habitats, crushing mussels, breaking tubeworm bushes, or burying carbonate structures. Accidental spills like the Deepwater Horizon disaster in 2010 release large quantities of oil and gas into areas that already host seep communities, and the interactions between natural and anthropogenic hydrocarbons add layers of complexity to understanding ecosystem responses. Research following the Deepwater Horizon spill has examined how seep communities responded to the influx of oil and gas. Some studies found that certain seep organisms, particularly those with methane-oxidizing symbionts, were able to utilize the methane from the spill as a food source, potentially helping to mitigate the environmental impact. Other studies documented negative effects on community structure and function. The full picture remains unclear, complicated by the fact that natural seepage rates are highly variable. making it difficult to separate anthropogenic effects from natural variability. The management of human activities in areas containing cold seeps requires balancing multiple objectives. There are economic interests in extracting hydrocarbon resources. There are scientific interests in studying unique ecosystems. There are conservation interests in protecting biodiversity. And there are practical interests in maintaining ocean health and ecosystem services. Achieving this balance requires understanding what activities are compatible with seep conservation and what measures can minimize impacts. Some jurisdictions have begun to designate cold seeps as protected areas or essential fish habitat. In the United States, certain seeps along the Atlantic and Pacific margins have been identified as areas where bottom-disturbing activities should be restricted. The logic is that seeps provide unique habitat values, support commercial fish species, and harbor endemic fauna that merit protection. However, enforcement of these protections is challenging in the vast and remote deep ocean, and monitoring compliance requires resources that are often limited. International waters, beyond national jurisdiction, present additional challenges. Much of the global ocean floor lies in areas not governed by any single nation, and the legal frameworks for protecting ecosystems in these regions are still developing. The United Nations is currently negotiating a treaty on biodiversity in areas beyond national jurisdiction, which would provide mechanisms for establishing marine protected areas in the high seas. Cold seeps, as unique and vulnerable ecosystems, are among the features that might warrant protection under such a treaty. Beyond extractive industries, cold seeps are relevant to other human concerns, including climate change, the carbon cycle, and the future of energy systems. The methane stored in hydrate form beneath the ocean floor represents both a potential energy resource and a potential climate hazard. As mentioned earlier, extracting methane from hydrates could provide fuel, but doing so safely and sustainably is technologically challenging. Meanwhile, natural release of methane from destabilizing hydrates could contribute to atmospheric warming, creating feedback loops that accelerate climate change. Understanding the dynamics of methane hydrate systems, including the role of cold seeps in regulating methane release, is critical for predicting future climate scenarios. Scientists use observations from seeps, measurements of methane flux, and models of microbial consumption to estimate how much methane is likely to escape from the seafloor under different warming scenarios. These estimates feed into larger Earth system models that project future climate conditions and inform policy decisions about greenhouse gas emissions and climate mitigation. Cold seeps are also relevant to the search for sustainable solutions to environmental problems. The microorganisms living at seeps are adapted to consuming methane and other hydrocarbons, skills that could be harnessed for bioremediation of polluted sites. Researchers are investigating whether seep bacteria could be used to clean up oil spills or to treat industrial waste containing hydrocarbons. The enzymes these bacteria use to oxidize methane might have applications in biotech or industrial chemistry. Exploring the practical applications of seep microbiology is an emerging field with significant potential. The education and outreach surrounding cold seeps help raise public awareness of deep-sea ecosystems and the importance of ocean conservation. The images and videos from seep sites are visually striking, featuring otherworldly landscapes and bizarre organisms that capture the imagination. When people see tube worms, brine pools, and bacterial mats, they're confronted with the reality that life exists in forms and places far beyond everyday experience. This can inspire wonder, curiosity, and a desire to protect these hidden worlds. Museums, aquariums, and educational programs use cold seeps as examples of extreme environments and evolutionary adaptation. School curricula incorporate lessons about chemosynthesis as an alternative to photosynthesis, broadening students' understanding of biology. Documentary films and online videos bring seep ecosystems into living rooms worldwide, making the deep ocean accessible to audiences who will never visit these places in person. Through these efforts, cold seeps contribute to ocean literacy, the public understanding of ocean systems, and the human relationship with the sea. Artists have also found inspiration in cold seeps. The visual aesthetics of tubeworm bushes, the eerie beauty of brine pools, and the conceptual richness of chemosynthesis have influenced photographers, painters, sculptors, and installation artists. Some artists collaborate with scientists to create works that blend art and science, using creative expression to communicate scientific concepts or to evoke emotional responses to deep-sea environments. These artistic interpretations can reach audiences that scientific papers and technical reports cannot, fostering connections between human culture and the natural world. The philosophical and existential dimensions of cold seeps are worth considering as well. These ecosystems operate according to principles that are fundamentally different from surface ecosystems, yet they're part of the same planet, the same biosphere. They remind us that our familiar world, the world of sunlight and plants and animals, is not the only way life can be organized. There are parallel worlds, hidden realms operating on different energy economies, and they've existed for as long as or longer than the surface ecosystems we know. This realization can be humbling. It suggests that human perspectives on life are shaped by our particular circumstances, our dependence on photosynthesis, our need for oxygen, our adaptation to temperatures and pressures at Earth's surface. Other organisms have different perspectives, different needs, different ways of being alive. The bacteria at cold seeps don't know or care about sunlight. They've never experienced it. Their entire existence unfolds in permanent darkness, powered by chemistry we would find toxic. Yet they thrive. They reproduce. They evolve. By any meaningful definition, they are successful. Understanding this can foster a sense of humility and connection. We are not the measure of all things. Life is more diverse, more creative, more resilient than any single species can fully comprehend. And the Earth is more complex, more layered, more full of hidden wonders than we often acknowledge. Cold seeps are just one example, but they exemplify a broader truth. The world exceeds our grasp, and there is always more to discover. And now, as we bring our journey through the cold seeps to a close, it's worth pausing to reflect on what these places mean. They are, in a literal sense, windows into the deep Earth, places where the interior of the planet meets the ocean, where ancient carbon stored in sediments is returned to the biosphere. They are laboratories of evolution, where life has adapted to conditions that seem hostile, even impossible. They are participants in the climate system, regulating the flow of greenhouse gases. They are ecosystems of extraordinary biodiversity, home to species found nowhere else on Earth. But perhaps most profoundly, cold seeps challenge us to expand our definition of what constitutes a livable environment. For most of human history, we've thought of life as something that requires sunlight, either directly or indirectly. We've built our understanding of ecosystems around the concept of photosynthesis, of solar energy captured and transformed into the chemical energy of food. Cold seeps remind us that photosynthesis is just one pathway, that chemical energy can serve the same function, that life is fundamentally about harnessing energy gradients wherever they might be found. This has implications far beyond biology. It shapes how we think about the origin of life, about the conditions under which life first emerged on early Earth. It informs the search for life elsewhere in the universe. It reminds us that life is resilient, adaptable, opportunistic. It doesn't wait for ideal conditions. It takes what's available and makes it work. You are part of this story. The atoms in your body were once part of other organisms, other ecosystems. The carbon in your cells has cycled through the biosphere countless times, perhaps even passing through a cold seep somewhere in the distant past, processed by chemosynthetic bacteria, before being incorporated into a tube worm or a mussel, which eventually died and decayed, releasing nutrients that fueled other organisms which were consumed by others, and on through the millennia until those atoms became part of you. The iron in your blood, which carries oxygen through your body, was forged in a supernova billions of years ago. It drifted through space, condensed into the planets, was incorporated into Earth's crust, cycled through geological processes, dissolved in seawater, absorbed by organisms, and eventually made its way into your body. You are literally made of stardust, but you are also made of the ocean, of deep time, of the endless cycling of matter through Earth's systems. The connections run deeper still. The proteins in your cells, the DNA that encodes your genetic information, the metabolic pathways that keep you alive. All of these have evolutionary roots that extend back billions of years to the earliest microorganisms. Some of the same enzymes used by bacteria at cold seeps to oxidize hydrogen sulfide or methane have molecular relatives in your own cells, performing different but related functions. The fundamental chemistry of life is conserved across vast evolutionary distances. You and a methanotrophic bacterium at a cold seep are distant cousins, sharing a common ancestor that lived in Earth's ancient oceans. This biological unity is profound. It means that studying organisms at cold seeps tells us something about ourselves, about the nature of life, about the possibilities and constraints that shape all living things. The adaptations that allowed tube worms to transport both oxygen and hydrogen sulfide in their blood are variations on the same hemoglobin theme that allows you to breathe. The symbioses between bacteria and their animal hosts at seeps are examples of the same cooperative strategies that have shaped your own evolution, your own microbiome, your own dependence on partnerships with other organisms. Even the energy transformations occurring at cold seeps parallel those in your own body. You oxidize organic molecules with oxygen to extract energy. Seep bacteria oxidize methane or hydrogen sulfide to extract energy. The chemical details differ, but the principle is identical, capturing energy from redox reactions, from the movement of electrons between compounds at different energy levels. All of life, from the smallest bacterium to the largest whale, operates on this principle. We are all, in essence, chemical systems harvesting energy gradients to maintain order against the inexorable pull of entropy. Tonight, as you drift towards sleep, imagine yourself descending through the water column. Imagine the surface falling away above you, the sunlight fading. Imagine passing through the twilight zone, where the last glimmers of light disappear. Imagine the water growing colder, darker, quieter. Imagine the pressure building as you sink deeper, thousands of meters below the waves. And imagine reaching the seafloor, a place of perpetual darkness, silence, cold. And imagine coming upon a cold seep, a place where the earth exhales methane, where bacteria convert chemicals into life, where tube worms and mussels thrive without ever seeing the sun. Feel the current moving gently past you. See the bacterial mats rippling on the rocks, white and orange films of microscopic life. Watch the methane bubbles rising from the sediment, tiny spheres wobbling upward into the darkness. See the tube worms swaying, their red plumes extended, filtering chemicals from the water. Notice the mussels clustered around seep vents, their shells packed tightly together, each one harboring billions of bacterial partners in its gills. Observe the crabs picking their way across the seafloor, the small worms burrowing through sediment, the fish hovering in the water column. This is a world powered by chemistry, by reactions between methane and oxygen, between hydrogen sulfide and sulfate, between compounds at different oxidation states. It's a world without sunlight, without photosynthesis, without the familiar energy pathways we take for granted. Yet it's alive, vibrant, productive. It's been here for millions of years, functioning according to principles that remained hidden until very recently. And it will continue long into the future. As long as the Earth continues to produce methane, as long as tectonics continues to crack the seafloor, as long as bacteria continue to oxidize chemicals for energy, you are part of the same chemistry. Your body too converts chemicals into energy. Your cells too are powered by metabolic reactions. You breathe oxygen, consume food, produce carbon dioxide. The biochemistry is different, but the principle is the same. You are a chemical system, capturing energy gradients and using them to maintain order, to grow, to repair, to live. In that sense, you are kin to the bacteria at cold seeps, distant cousins separated by billions of years of evolution, but united by the fundamental challenge of staying alive. Rest now. Let the images of cold seeps fade gently from your mind. Let your breathing slow, your muscles relax. Let yourself sink into sleep the way an ROV descends into the deep ocean, steadily, peacefully, into darkness. The cold seeps will still be there tomorrow, pulsing quietly on the ocean floor, exhaling methane, supporting life, connecting the deep earth to the ocean, the past to the present. And you'll still be here, made of atoms that have journeyed through the cosmos and through the earth, part of the same grand cycling of matter and energy that includes cold seeps and stars and everything in between. If you're still awake enough to move, consider subscribing so you don't miss the next journey into the ocean's mysteries, and perhaps leave a comment letting me know where you're listening from tonight. But if sleep is taking you, then go gently. Dream of the deep. Dream of life powered by chemistry, of ecosystems in darkness, of the endless creativity of evolution. Dream well. Good night.